And with that, please join me in welcoming Professor Olson to his lecture, Grasping the Molecules of Life, Modeled in 3 Dimensions. Professor Olson.
Thank you. Well, thank you very much for that long introduction. And particularly like to thank the Philosophical Society for inviting me first and making me a member second. And especially Larry Milstein, who gave the invitation. So what I'm going to talk to you about today is how we understand and visualize and explore complex molecular systems that underlie the processes of life. Before I start, I want to acknowledge the people. Actually, if you could keep the lights up, I think the slides are probably good enough to see. First, I'd like to thank Graham Johnson and Ludovic Autain, who did a lot of the work that I'm going to show later in the talk about cellular environments at the molecular scale, and 2 very long-time associates in the laboratory, Michelle Sanner and David Goodsell, who have been both inspirations and contributed greatly to the work of the lab, and also to some technicians whose work physically you will see, Adam Gardner, John Huntoon, and Max Nanis. And the rest of the molecular graphics lab, which works highly interactively to develop these kinds of projects. So really, I just want to make 3 simple points about interacting with the molecules of life. For humans to understand things, they have to perceive them. And we should think about perception as more than just using our eyes, because we learn in lots of different ways. As far as the data that we need to understand these life processes at the molecular level, we have to think about integrating data across scales. So we need to synthesize information that comes from different sources into a single picture. And finally, the tools that we use are computational tools, and what that means is that we have to develop software. The trick here is not to reinvent the wheel, but rather to try to reuse as much as we can in the context of the domain that we're studying. So we need to bridge disciplines rather than reinvent things that have already taken place and been developed. So the important point that I'd like you to think about is that we live in what I call the age of bioatomics. And that means that we now understand that there is a physical continuity from the atom to the organism. So concepts in some sense like mind and body disappear. Mind and body are one and the same thing because they both reflect the physical nature of living systems, and in this sense, conscious living systems. So the question is, how do we get to understand this type of complexity? Well, first of all, we know that the laws of physics are still obeyed in the biology that we study. And the chemistry that we know still takes place. But there's a complexity that's introduced in chemistry that is not present in the same way in physics, and that's the chemistry of combinatorics. So you have, let's say, 92 natural elements and the transuranium elements. They can recombine in lots of different ways. That combinatorics gives you a complexity that you could not predict from the physical laws itself. In fact, if you just take the elements that are in your body, let's say the 5 or 6 most prominent elements, so carbon, nitrogen, oxygen, hydrogen, phosphorus, you can make 10 to the 60th different chemical compounds. That's enough to fill up the entire universe. So that kind of complexity needs to be dealt with. Now, when we're talking about biology, there's an even greater level of complexity, and that is the complexity of these chemical entities interacting in an environment and responding to the environment by encoding the history of that environment in how they evolve. That is an incredible amount of complexity, and you'll see that the types of molecules that have evolved over the 4 billion years of the evolution of living systems on the Earth is an amazing amount of complexity. Our goal is to uncomplicate that complexity. What we want to do is to understand it. In order to understand it, we need to uncomplicate it, not reduce the complexity, but break it up into meaningful pieces. I'll just mention this image on the left. Some of you may have seen it before. It's a watercolor painting that was done by my colleague at Scripps, David Goodsall, and it represents the molecular environment of a piece of the E. coli bacteria. And like I say, this is watercolor that is based on the known shapes and abundances of the different molecules in that environment. So I just want to briefly show you what kind of scale we're talking about. So these are things that we can see with our naked eye, a coffee bean and a grain of rice. But as we go smaller, we start to see things like amoebas and single-celled organisms. If you look in the left, we're going up scales of 10 for each one. As we go down, we see smaller cells, red blood cells, yeast cells, structures, subcellular structures like chromosomes, mitochondria, lysosomes. This is that bacteria that David illustrated. And as we go smaller yet, we start to see things like bacteria, measles, virus, and smaller bacteria, the influenza virus, HIV, hepatitis virus, coated vesicles. And then as we go even smaller, we go down and start to see individual proteins. So these are hemoglobin, tRNA, antibodies, phospholipids. We start to see things like, let's go back a second, these things, methionine, which is one of the amino acids. There are 20 amino acids that make up proteins, and down to a single carbon atom. So these are the levels of complexity that we're dealing with. How do we understand that complexity? How do we get information about these different levels of structure? And the answer is, there is no one way. There are lots of different ways that we need to get data at different scales. So we can use light microscopes to look at individual cells, but when we start to go down to molecular assemblies, we need things like electron microscopes and tomography, electron tomography. If we go further down, we need technologies that use higher frequency wavelengths, shorter wavelengths, X-ray, to get atomic level structures. So these are different experimental modalities that we need to combine in some way to get a broad picture of these complex systems. So just a little bit of background history. This is a picture of Linus Pauling, who is probably the best known and one of the best structural chemists of the 20th century. And he used X-ray diffraction to study the shapes, the atomic structure of minerals, principally. But he understood the power of visualization and developed physical models so that he could actually understand the three-dimensional relationships of these atoms in these structures. In fact, he used 3D models to predict structures that were not even known. The structure on the right is actually an alpha helix, which is a component of a protein. And he basically one day just started to fold paper to try to make these hydrogen bonds that enable the protein to fold up, and he discovered a motif that he called an alpha helix. This was before there was any experimental data on that structure. It turns out he predicted it 10 years ahead of the time that it was actually seen experimentally. This next case is something that you're all familiar with, which is the Watson and Crick model of the DNA double helix. And I'm sure many of you have read the memoirs of both Jim Watson, the double helix, and Francis Crick, which was called What Mad Pursuit. In it, they talk about how they were able to take Rosalind Franklin's data, which is shown on the left, which is a fiber diffraction, again, using X-rays of fibers of DNA, and interpret that in a physical model. And if you read the memoirs of these two scientists, they both talk about how important it was for them to physically play with the model. So they had a machinist make these nucleotide base pairs at the right scale to each other, and they were able to develop this three-dimensional model because they were able to use this model essentially as an analog computer to develop a consistent picture of the distribution of atoms that caused that diffraction pattern. The interesting point that I found out just a couple of years ago when I went back and I read the Nature paper, the 1953 Nature paper, was that they did not mention the physical model at all in that paper, which is kind of a shock if you think about it, because it was so central to their discovery. And then you ask the question, well, why didn't they mention it? And the answer is that scientific papers don't typically talk about how you get ideas. They talk about how you prove ideas. And so a lot of science is hidden under the rug in some sense, which is how is it that scientists understand what it is that they're trying to understand? So this is something that I became pretty interested in, and it kind of led to some of the work that I'll show you in a moment. Things changed radically in the early to mid-1970s when three-dimensional computer graphics came around, was developed. And a lot of people don't appreciate the fact that the technology that you have on your cell phones now, 3D graphics, the technology that you watch in the movie theater, 3D graphics, had to start as a startup. These machines in the 1970s were very expensive. I bought my first computer. It was the first computer to be bought for research at the Scripps Research Institute when I first arrived there in '81, and bought a computer graphics system. The whole thing together cost over $500,000. You have in your pocket probably a smartphone that has better graphics, more processing power, more storage, many times more storage, and you get it with your phone plan now. So things have changed radically, and it certainly changed the way we look at models. So the one point that I want to make here is that the computer has expanded our capabilities incredibly, but every time you adopt a technology, you also leave something behind. And in some sense, what the computer did was dematerialize the physicality of these structures that are real physical structures, only too small to perceive without a great deal of technical aid. So biological structures are very complex, and the spatial relationships and reasoning is very difficult. And the typical kind of interface that we use for our computers, mouse and keyboard, are quite limiting. And in fact, images, even 3D images, even stereoscopic 3D images, may not convey the entire picture. So I started to, about 15 years ago, started to look back kind of nostalgically at the physical models that I first built when I started in the field, and also took a look at some of the social trends and technical trends that have evolved in the meantime. So video games and the iPhone and iPad have all brought interaction embodiment back into computing. People now, I saw a video the other day of some baby trying to swipe his hand across a magazine to change the picture. So things get pretty complicated when a gesture that is meant to mimic a physical action, it now replaces a physical action in a physical object. But the fact is that the embodiment and the way we interact with things has changed dramatically. And the fact is that also computers are in everything. Computers exist, they're very, very small, and they exist in just about everything that we use. Video is also ubiquitous. And a new kind of video is coming along, which is basically depth video, which actually not only gives you a picture on a flat screen, but gives you three-dimensional information about the scene. So for $150, you can buy a Kinect box that goes with your Xbox that actually allows you to get three-dimensional information about your interactions. So usually they're using it for dancing games and so forth. But what it really does is connect the real world in a different way with the computer world. And that goes along with the development of what's called augmented reality technology. So augmented reality technology is the technology that allows you to integrate the physical world and the computing world in a seamless way, and there are lots of ways of doing that. The other technology that's evolved over the last 15 or so years is computer auto-fabrication, sometimes called 3D printing. So now, instead of printing a picture, you can print an object. And you can print just about any object, as long as you can build it in a computer and it has good physical characteristics. So the goal that we've set out is to use these technologies to provide natural computer interfaces for exploring and learning about these complex molecular structures. So there's no reason that we can't think about designing a specific interface for a specific problem now. You just print out your interface that has the physical characteristics that you want, and you use that to guide computation or exploration. For those of you that don't know about these technologies, these 3D printing technologies, they're called additive manufacturing technologies. So basically, they build up objects on a layer by layer, with a layer by layer pattern, until you have a full 3D object. And there are technical aspects to how you support things that don't exist at the layer below but then appear above and so forth. So there's support issues and so forth. And that has a lot to do with the kinds of materials that you use and so forth. So some use powder-based systems where you basically spray color binder layer by layer and build this thing up in the context of loose powder, and then you kind of dig it out and dust it off, and then you've got your object. Some of them kind of just build with two materials. One of them is a support material, and the other one is the model material, and that you can remove the model material, the support material, through dissolving it and so forth. Currently, there are probably a dozen different materials that you can print with that range from plastic to titanium. And there are lots of mechanisms. You can not just make a monolithic piece, but you can make things that actually have mechanisms involved. So why do I think that physical models are important? Like I said, I think that the perception itself, when we perceive something, we perceive more than just what we're looking at. We perceive things as we hold them, as how they exist in the world and so forth. So physical objects engage more than the eye. They engage the bodily senses, tactile senses, proprioception, and so forth. And they also provide a kind of perceptual coherency between these different things. You've got a real object there, and so it is coherent in terms of manipulation. You know, it's not going to fly off into infinity like sometimes bad code makes a object fly off the screen. But also, it's from a cognitive point of view, it provides an intuitively and intelligible object. In other words, you understand it immediately as a physical object. So if we're talking about the physicality of something, then having a physical object is intuitive. You don't have to think about moving a mouse in one direction to turn it around. If you want to see the other side, you just turn it around. If you want to look closer, you just look at it closer. So it's contextual as well as manipulable. Also, like Watson and Crick showed, that you can use it in an analogical way. It's an analog computer in some sense. It has characteristics. It isn't the molecule. It isn't the molecule itself. It's a model of the molecule that represents certain characteristics. But those characteristics have meaning. And so you can use that meaning in an analog way and in a metaphorical way. And in addition to that, there's a certain social aspect about physical objects. They're much more social than a screen. You can share an object. It's persistent. You have it in on your desk. You can go back to it and look at it and talk to people that come in. We use it all the time in, let's say, our HIV work when we're having a discussion about sites of resistance evolution on a particular molecule. Having a physical model and pointing to it and talking about it in a physical sense is intuitively easy for people to understand. So all of these things, I think, go beyond just the visual. So what I'd like to do is to show you, if we can get this to work, a couple of demonstrations. Okay. So this is a model. This is a physical model of a virus. This virus model is based upon the atomic level structure of the polio virus that was solved back in 1985 by a colleague at Scripps. But it's not just a monolithic model. It's a model that can come apart, because viruses have to come apart to leave a cell, and then they have to put themselves back together. Now, who puts them together? Well, it's just Brownian motion. So if I shake this, you can see that this thing is starting to assemble. Now, why is it assembling? Well, it's assembling for two reasons. One of them is that the shape is based on the shape of the virus. And in addition, I've made an analogy between magnets on the edges of this model and the electrostatic complementarity of the edges of the virus at a different scale. So as I shake it, you start to see it come together. Now, you know that, in fact, what's called the lowest free energy of the system is when all of the bonds, the possible interactions, are made. Now, there are 12 pieces here, and some of them still have open bonds, magnets that haven't found their mate. Now, I'm just shaking this randomly, so I don't know how long this is going to take. They said I could talk for as long as I wanted. But one thing I know for sure is that eventually it will come together. While I'm shaking it, I'll tell you a story, because I took a video of this and I put it on YouTube. And you know how everybody likes to comment about your videos on YouTube. So when one of the first comments, well, let me first say that I shook it. When I took the video, it went together in about 30 seconds. But you know, you can choose how long it takes if you shake it a number of times. At any rate, the first comment I got was, "Well, you just broke it apart and then ran the video backwards." So then I thought I was going to be real smart. I made two different models, one of them red and the other one green, but they were chiral opposites. They had left and right-handed. So it meant that the green and the red couldn't bind together. And then I put them in a rock tumbler and let them tumble. And I put that movie on YouTube. And believe it or not, I thought it was being smart. So I put my wristwatch in the video so that you could see that the time was going forward. So what was the first comment that came back? "Oh, you just made your watch run backwards." So in fact, that's an encouraging thing for me, because it is counterintuitive to think that you can actually assemble, this one's taking longer than usual, but that you can actually assemble physical models in just by shaking. And in fact, you know, I've had 3-year-olds shake this much better than I can do it, obviously. At any rate, I won't. You can. Sorry. The lower temperature. Lower the temperature. The temperature? Lower the temperature a bit. Keep going. Okay. So the thing is that the amount of shaking is analogous to the temperature. It's the kinetic energy that you're putting into the system, right? So if you shake it too hard, it will never come together, because the energy is higher than the free energy of the assembly. If you don't shake it hard enough, it'll never come together. So in fact, you have to shake it kind of at the right temperature. So let me show you. I'll move over to another. So we've been using this in schools. Let's see. And so what we wanted to do was not to throw away the computer, but to use these technologies of augmented reality. So here, for instance, I can identify the object by its color. So I'm just thresholding it on the yellow color. If I can do that, I know its position. And if I know its position, I can track it. Now, as I track it, as I shake it, I can see what the temperature is. If I shake it too hard, it falls apart. If I don't shake it hard enough, it doesn't come together. If I shake it at the right level, it should go together. So there's another. So you can teach a fair amount of physical chemistry in this, because now this thing is almost totally assembled except for one spot. The problem is that, actually, we've got another phenomenon here that you can talk about. One of them is trapped inside. Now, that actually is a known chemical phenomenon. It's called a kinetic trap. And what that means is that if you want the thing to assemble fully, then you have to raise the temperature to break it and try again. And that's what nature does. It doesn't just go in one direction. It goes until it can actually reach an optimal structure. So let me show you just a couple of other physical objects that I've been developing in the same vein. So let me switch back here for a second, if I can. Okay, so this is another model. This is a model of a peptide chain. It is the backbone of all the proteins that are in your body. And this peptide chain folds up because of certain interactions. These interactions are called hydrogen bonds, and these are printed on the 3D printer with magnets that represent the donors and acceptors of the hydrogen bond. So I can take any number of these, put them together. Let's see if I can do this right. And I get what's called a beta sheet. And if I fold them up, I get what's called a beta barrel. So these are common folding motifs of proteins. So proteins actually put themselves together because of the geometry that is embodied in this model. Now, let's say I want to put some side chains. So this is just the backbone of this long polymer, but there are 20 different kinds of side chains that go on this polymer. So again, I can use augmented reality, and here is a simple side chain that is basically generated on the computer because this marker is recognized, and then when it is recognized, there's a pattern inside that says this should be, let's say, a glycine, or let's take another one. So now what you have is a hybrid model. You've got a model. So this particular side chain here is actually cycling through all of the different 20 amino acids. So as I manipulate the model, I can manipulate these side chains. But more important than that, as I manipulate the model, I'm actually manipulating the model. Oh, let me put this on here. Okay. So here I'm manipulating the model in the computer, right? So that means that I can calculate the distances between these side chains. As I rotate this side chain, let me get a black background here so you can see it. As I rotate the side chain, we need the right, oh, sorry, this is, yeah. Yeah, here we go. Okay. So the technology basically depends on this lighting from the video. And let me get a little bit closer. Hang on, let me try a different one. So it needs to recognize the pattern on the inside. Okay, here we go. So now, as you see, if I rotate the side chain, it changes the distance. So if you know the distances between atoms and you can manipulate them on the physical model, you can calculate the energies of the interactions and so forth. So the technology, although it's prototype at this stage, actually has a lot of potential for lots of different applications. A couple of different applications, just so that you see that it's quite general, is here. What you're looking at are the different FDA-approved drugs that fit into the active site of an HIV drug target, which is called the HIV protease, which is one of the molecules that we study in our NIH program project. And so it's cycling through these FDA-approved drugs in the physical model. A couple of things that you might notice. Let's see if I bring this over here. Is that the physical model is occluding the computer graphic model. And that's because this physical model is made on a 3D printer, so it's really, I have the model in the computer to begin with, so I can use it as a mask against the physical model. In addition to that, I can use augmented reality to do kind of other types of exploration. So here, I'm using it to look at the specific interactions around the HIV. So these are technologies that exist in augmented reality, and it turns out that augmented reality is now becoming commonplace, just like 3D graphics have become commonplace, especially on mobile devices. I'll show you one other example. So this is a model of a protein called superoxide dismutase, and what you're seeing. Let's get the tracking done here. Okay, so what you're seeing here is this cloud around the physical model is the electrostatic potential that surrounds this particular protein. And this protein uses that electrostatic potential to attract reactive oxygen species, negatively charged superoxide, into this active site, which is where my finger is pointing right now. And in fact, these little marching ants that you see are the directions of the field lines around the physical surface of the protein that where a negative charge would actually follow those field lines into the active site, which then dismutes the reactive oxygen species into something that the body can actually handle, so it won't do damage. And in fact, you can have two of these if you want, and each one has its own electrostatic field. So using the physical model and using the physical models both, you can try to get both the shape complementarity and the electrostatic complementarity. Red wants to, which represents negative charge, wants to be close to the blue cloud, which represents positive charge. So these are technologies that we're trying to develop now, both for the purposes of teaching and for the purposes of exploration. So let's go back to the now. These are, I always have a backup for a live demo because they don't always work, so I can skip these now. So now I want to step away from these individual molecules and start to look at the complex environments in a cell itself. And like I said before, it takes the integration of information from lots of different sources in order to get a picture of the cell. And Graham Johnson, inspired by the work of David Goodsell, who did those watercolors that I told you about, as 2D illustrations, came to the lab as a graduate student with the goal of trying to make this into an automated 3D process where you could actually take information about the shapes and the types of proteins that are in a cellular environment, the copy number, how many of each there are, and how they fit both on the surfaces and in the interior spaces, and put them together into a picture of a cell. Now, Graham came to the lab after being a scientific illustrator for 10 years to get his PhD in molecular biology. So he came from an artist background. And I think that combining the aesthetics and the understanding of how we see things from the arts is a very important part of developing good scientific visualizations. This is a movie that he made recently showing things at the cellular level and how you might want to clarify certain aspects of cellular structure. I'll play this.
This rotating 3D model shows a single cell similar to the trillions of cells that make up your body.
Could you hear that?
This particular cell came from the pancreas of a mouse, and it's used as a biological model to study diabetes in humans. Fading the outer membrane unveils the cell's internal organs, called organelles. The round insulin-secreting vesicles, color-coded here in blue, dominate the interior. 12,128 of them in total. Fading these blue vesicles reveals a network of mitochondria, color-coded in green, sprawled about the yellow, color-coded nucleus. Scientists and general audiences alike can learn a great deal about biology by comparing internal structural differences between cells harvested from different environments, that is, from different parts of your body or from different life cycle stages. But even comparisons that seem obvious in a single model can deceive, let alone across multiple models. For example, approximately how much of the cell's total volume do you think that this yellow nucleus occupies? About 12%, 26%, or about 39%? How much of the cell's total volume do you think that the green mitochondria occupy? About 7%, 42%, or about 68%? Believe it or not, the correct answers are closest to 12% for the nucleus and only 7% for the mitochondria, much lower than most people infer by glancing directly at these representations of the data. A tool we've developed allows scientists and general audiences alike to compare and contrast multiple descriptive parameters of visually complicated 3D structures, like this cell, at a glance. By organizing the organelles and averaging their volumes, we can represent the cell as a simplified 3D graph that retains qualities of the native model. Here, a viewer gets a gut-level impression of the relative volumes of each of the organelles, far closer to the correct values, but they can still appreciate that this data represents information about a cell rather than something more mathematically abstracted. The same tool enables us to rapidly compare otherwise similar-looking cells. The two cells on the left, collected after glucose stimulation, are smaller and have a different organelle distribution than the two unstimulated cells on the right. This information is much easier to absorb by looking at the simplified cells on the bottom row, rather than by mentally merging the information from the original models in the top row with the quantification information in the pie chart shown in the middle row. After highlighting comparisons with the simplified information, the cell can morph back to its original shape, reminding audiences of the complex interplay of randomness and specific interaction in these structures that enables life to exist.
So this data was actually generated experimentally by a scientist in Australia, Brad Marsh, and it took a long time. He actually had to mosaic thousands of tomographic slices of these insulin cells in order to get this data. And the data itself only shows things at the organelle level. It doesn't show the molecules themselves. So the goal was to take data like this, that's captured at one scale, and integrate it with other data that's captured at a finer scale, like the paintings of David Goodsell. I had worked on that problem a little bit about 15 years ago and generated this image of a blood serum with antibodies and a virus in it. It took me about 3 or 4 weeks to generate this three-dimensional model from all the data. When Graham came to the lab, he was expert in some of the high-end computer graphics animation software, and he thought, well, all we need to do is kind of stick these number of these things into a volume, run a collision detection, and squeeze it down to the density, the correct density of these objects in the cell. It turned out that that was very computationally expensive, and it could work with blood serum, which is 1/5 the density of proteins that you find inside the cytoplasm of a cell. So that was not the solution. On the other hand, the problem itself, the packing problem, it's called a packing problem, and it's been studied both in mathematics and computer science, but usually with abstract objects or just simple growth algorithms. So the image on the lower left is a packing of different size spheres that can be solved mathematically. On the upper left is a tree growth algorithm that was developed several years ago. But nobody has actually put all of this together into a packing problem where you have all of these different shapes residing on different organelles, inside different organelles, on membranes, and in cytoplasm, like the image on the right that David Goodsell painted. So Graham went ahead and devised some algorithms in collaboration with Ludovic Autain and Michel Sanner to develop a scheme in which he would take known structures at the higher level, like these organelle structures represented by the sphere and this tube, like a nucleus and a mitochondria, and some large molecular assemblies that are represented by these fixed P's, and embed them into what you might think of as a smart graph, something that knows where everything is at every point around it. And so, I want to go back a second. So basically, if you have a recipe of the proteins, the objects that pack into this volume, you can at least recapitulate the densities of these objects in the right places. So, for instance, in this case, the yellow cube type objects are in the outside cytoplasm. The green cubes and triangles are inside of one type of organelle, and these ellipsoidal pink shapes are inside another organelle. So it's just a packing of geometry in some sense. But not all biological molecules are compact. There are several molecules that are fibrous types of molecules, very long, thin, like actin fibers and tubulin and microtubules and so forth. Lots of proteins exist on the membranes that separate different organelles from the cytoplasm and from each other. And so you need a generalized set of algorithms that can put these things together, and that's essentially what Graham has developed. In addition, you may have information about gradients, different concentrations of different objects as they exist in relationship to other objects. So all of these things have been put together into a set of software that we call autofill, which can fill up space, different types of spaces, different surfaces, and have procedural elements that can make long, thin objects into tightly packed environments. And then once you have built that, you can start to query it by removing and looking at different things in different ways. So developing these models requires a variety of different approaches. Here, what you're doing is growing a long, thin fiber like DNA with a certain so-called persistence length, in other words, that can bend a certain amount but not kink, and you fill it up. You fill up between existing structures. So all of these algorithms have been put together into autofill, and then when you add the biological information, it becomes what we call autocell, which can fill up cellular spaces with all of these structures automatically. So you can use that and develop analytical tools for the model itself. So for instance, this heat map, or this red to white mapping, is showing the distance of each one of these molecules to a particular fiber fibrous molecule in a cell. So you can start to analyze these models once you've built them. So again, starting from the early work of David Goodsell and myself, Graham has gone to being able to automatically, what took me like 3 weeks, this was done in about probably 5 seconds using these automated techniques. In fact, I'll show you. So just to show you, again, that blood serum, here we're focusing in on blood serum. This is a structure of high-density lipoprotein. So this is the good cholesterol that you have in your blood. And this is blood serum. This is 1 micron across of blood serum filled up with all of the proteins in that cell. And that was done automatically in probably 2 minutes, a minute or two. And most of that time was spent in the rendering. And here's another example of an early example of autofill of data that was published in 2006 of a synaptic vesicle. So these are all of the proteins that are on a synaptic vesicle. This is the carrier of the neurotransmitter that goes from one nerve cell to another. Now, the third component that I want to talk about is reusing software. As molecular biologists and computer technologists, we've developed a lot of software for doing molecular modeling. So the software that we have is three-dimensional, it uses 3D graphics and so forth, but it focuses on the scientific domain. So this is not just gives you a 3D representation, but each of those geometries have meaning, have syntactical meaning in terms of what they are. So if I want to color it a certain way, if I want to show only a certain type of amino acid, if I want to do these things, I can do that in my molecular modeling software. Now, in the meantime, computer graphics has moved way far ahead because it's driven by the game industry and by Hollywood. So things that you see on the left, the kinds of animations and the kinds of collision detection that you see, or even if you have representations of proteins, you can do collision detection and they would bounce off, they could all bounce off one another. You can have thousands of these molecular structures bouncing off one another, but they don't know their molecular structures. They're just dumb geometry bouncing off one another. So on the one hand, you have the molecular modeling software that has the domain knowledge. On the other hand, you have these high-end computer graphic systems that can do all kinds of things very, very efficiently and quite amazing in terms of the physics engines that they have and so forth. So what we were able to do was because of some software capabilities and the way we engineered our software to begin with and the language that we used, it turns out that a lot of the high-end computer graphics software has an interpreter built into them called Python. The technical, you don't need to know anything about it, but we developed our software using that same interpreter. And so we can just move our molecular modeling information into the host software, this high-end computer graphics software, and have the best of both worlds with very little extra programming, because we just have some lightweight sockets to communicate between the two. So we can then use all of the computer graphics stuff that is available in the Hollywood animation, in the game industries, and we can use that with respect to all of the 3D graphics that we were able to do, like the augmented reality, interactive molecular dynamics, and actually using the augmented reality to manipulate the structure. That white ball is basically a force pulling the structure in a particular direction, or see different kinds of movements, dynamics at different scales using both the 3D graphics and the high-end and the molecular modeling. This is an example of docking, which was mentioned in terms of what we do. So here, you're trying to dock two things. In a molecular modeling package, those two things can pass right through each other, but with the computer graphics software, there can be collision detection, so they avoid one another. And in fact, each of these molecules can move out of the way with the collision detection, which is done in real time. And then we can use our molecular modeling software to compute the energy as we're moving these things around. So all of these things are made possible not by reinventing the wheel, but by putting software together in new ways. So that autofill, which was originally developed in a high-end graphic system that Graham was used to using, got moved over into the world of the molecular modeling, but then embedded into the high-end graphics. And so we're able to generate models very, very quickly and use high-end animation capabilities and rendering capabilities. Here's an example of the HIV virus as a 3D model that was filled up automatically, knowing the structures of the components of the virus itself. And this is an example, I mentioned these synaptic vesicles. So this is, again, electron tomography of a synaptic cleft. So the blue thing is a bouton from one neuron, and the yellow surface is the neuron across the synaptic cleft. And these white spheres are each a synaptic vesicle that hold the neurotransmitters that leave and go across that cleft. Now, with autofill and combined with the high-end graphic system and the molecular modeling, you can cut out a volume in the synaptic vesicle and fill it up. Here we're just looking at the lipids that form the membranes around each vesicle, and the green are the neurotransmitters. There's about 4,000 neurotransmitters per vesicle. Here you're showing one protein, and here you're showing all of the proteins in each one of those vesicles. So you can see how tightly packed and how complex these environments are. And finally, you add the cytoplasm, and you've got just this solid mass of proteins, even though it takes up about 50% of the volume. So what do we do with these models? Well, the fact is that once you build a physical model, you can run simulations. And so, and it turns out that the most time-consuming aspect of running simulations isn't the simulations themselves, because those are run on the computer. You just have throw more computers at it, and you can do more simulations. The time-consuming part is building the models, and building as many different models. If you want to test a variety of different hypotheses with different parameters, you want to generate these models very, very quickly. So that's what one of the areas that we think this technology is going to help in terms of actually simulating the environments inside cells at the molecular and atomic level. I'd like to close just by saying that visualization itself is the way science can be understood by everybody, by students, by the lay audience, and by other scientists. And certainly it's the fact that if we're worried about the future of science, we have to worry about our future scientists and make sure that we engage and educate them properly. And in addition, communicating this information, I think, is important to maintain a scientifically informed public. And with that, I'll close, and thank you for your attention. And happy to answer questions.
Please stand up, say your name, and also say you're a member of the society. Carl?
Hi, I'm Carl Merrill, I'm a member of the society. I thought it was spectacular what you presented, the ability for people to see this final part where you could build the physical aspect in, so you could get force interactions. And it raises a question in my mind, and I thought maybe you could comment on it, and that is the ability to use haptics, so that you could actually feel things as you get close.
So we actually have a couple of haptic devices in my lab, and you can program them. That, for people that aren't aware, these haptic devices have sensors and actuators. And it's like moving an object, and it's attached by a series of links with motors to the computer. So as you move the physical object, you can feed forces back into the arms. The problem with it is that they're still very expensive, and they also kind of get in the way. In other words, you have to tether this thing to a physical object, which is this long arm. There are people that have played around with a variety of different mechanisms, but there's nothing that I've been fully satisfied with. So, in fact, even though you don't feel the electrostatics here, you feel the volume exclusion. So you do have some tactile representation. And with these things, you actually feel not only the shape, but kind of the flexibility of the shape. So they give you a little bit of that haptic sensation, but it's not accurate, first of all. But the question is, it depends on how you use the model and what you want to get out of the model. The model is not the molecule. It only represents aspects of the molecule. So the more, I think one day, yes, haptics will come into it. It's going to be very difficult because, for instance, the only forces at a distance that we usually can feel are magnetic forces. But magnetic forces behave differently than electrostatic forces. The fall-off is much more rapid, r to the 6th instead of r cubed. So you can't exactly do that. On the other hand, because computers are getting small, actuators are getting small, I think it is going to be possible to use something like electromagnets that are displayed. There are these arrays of magnets now that are being developed so that it's not just a single magnet, but they're full arrays that you can control. So I think the technology is just beginning, and haptics is going to, I think, come into it more and more.
I also enjoyed the lecture very much, but I noticed that your projection of yourself was a mirror image of you.
Yes.
And you talked about symmetry in one of the slides. The question is, can you simulate the asymmetric models of things like thalidomide?
Anything. It'll take.
Thalidomide
Right.
babies were a result of a mirror image where the test had been conducted with
Yeah.
identical things, and they were half mirror image and half.
Yeah. So let me explain some aspects of this augmented reality technology. So there are lots of different ways that you can display the augmented reality technology. Now, because I'm sitting here moving and looking at the screen, so I'm looking at the screen as if it were a mirror. Okay. And because I'm doing that, I find it very difficult. I mean, I could just flip it back around so it was right and left, but as I move my hand this way, the object, I'm going to see the object go that way, which you can do. In fact, I just have a little switch there that turns on and off the mirror. And in fact, when we use it without showing it to an audience, typically we'll have the camera more from the viewpoint, from your own viewpoint. So what you're looking at is myself manipulating the model, and then in front of me is the screen showing the augmented reality. So there, the chirality or the handedness is the same. So it's just but I agree that it can be a point of confusion for people that are looking in this way, but you can have it the other way if you want.
What about simulating a chirality problem?
You can have right and left-handed, and I can have right and left-handed DNA. In other words, I'm not flipping, I'm not altering the handedness of the model itself. It's only the augmented reality that I flip just for convenience. Now, one might think that, in fact, in the not too distant future, there are some large companies working on these things. You could have glasses that integrate the real world with the computing world. Google has something called Google Glasses, and there are some other ones that are coming along. So in this case, again, the chirality would be exactly the correct chirality. You don't need to do this magic mirror trick. So it's just another way of making it easy for the human to. So if I look in a mirror, you know, if you tie, let's say you have some right-handed or left-handed tie, you know that it's a mirror image, so you just flip it. You know that it's just the other-handedness. So aside from, let's say, getting a student confused by showing a left-handed. So if I hold up a DNA model in this magic mirror mode, it won't be a right-handed DNA model. It'll be a left-handed DNA model, which obviously isn't correct. So I would have to explain ahead of time that this is a mirror. So you're looking at this right-handed model in a mirror, and it looks left-handed. Yeah.
That was an interesting error when you shook it and you had that trap state, little particle inside big particle. Does that happen in nature? And I don't see how it would fall apart since the temperature of the body.
Well, it doesn't happen with viruses, but it does happen with molecular systems. And so, for instance, in protein folding, there are proteins that fold up and get trapped in a non-native state. And they need to be basically unfolded. The fact is that in biology, there are lots of mechanisms that don't involve raising the temperature, but involve sequestering these molecules in different environments and having them refold. These are called chaperones. So chaperone is kind of the chemical equivalent or the environmental equivalent of releasing a kinetic trap. It basically provides a different environment that allows these things to unfold and then gives them another chance to fold. So, but in fact, these kinetic traps do happen. This is the simplest example where, in fact, you have to actually increase the temperature, in this case, or somehow break apart the existing structure using some mechanism to give it another chance to refold. In a sense, it's an error correction mechanism that exists across all of biology. In other words,
Scientifically, the error correction, this is scientific.
My name is Ivan Amato, and I'm a relatively new member. I know I pay dues. So these were spectacular visualizations, and I just, really excited to see these as tools that certainly must, help the imagination of the molecular scientists sort of figure out what's going on. But I want to go back to the beginning of your talk, where you were showing us tremendous sort of representations of scale, and also to your idea in the beginning that we go from atoms to consciousness in a biological context. So everything you showed sort of went from, say, cell down, that scale.
fell down meaning...
from the cell scale down to the atomic scale,
Mhm.
you're looking at neurotransmitter
Right.
molecules and vesicles. Question is, are you hooking up with other colleagues that are going from the cell up, so along that continuum to the full organism, to a brain, to perception, for example, where some of these kinds of brilliant visualizations might help
So-
navigate those categories?
So I am part of a collaboration with a number of scientists. It's called the National Resource for Computational Biology, and or Biological Computation, NBCR. And some of the scientists there are interested in heart mechanics. So they measure the fiber directions and the electrophysiology of hearts. They also measure the calcium fluxes in the cells themselves. So there are people that are working their way down from the top, and we're working our way up from the bottom, and we're trying to meet someplace in the middle. It's kind of like the Transcontinental Railway. You hope that eventually you're going to meet. There are examples. It's very difficult to find an example that goes where everything is known, enough is known continuously from the atom to the organ at this point. There are always gaps. I mean, what you learn in science is what we don't know, not what we know. That's the goal. And filling in those gaps is going to take some time. But there are certainly examples where knowing a molecular mechanism can have immediate impact on the organ or the organism. I mean, that's what drug design is all about. But you don't need to know everything, but it would help. I mean, the thing is that drugs are imperfect because we don't know everything that takes place in between. There are always side effects, there's always cross-reactivity, because we don't know enough about the system as a whole. So I think that the big change in at least biological science right now is the understanding that it's a system that we're looking at. It's not isolated pieces of the system. And the more we understand about the system, both from a structural point of view and from a kind of a network point of view. And I think that the real progress is going to come when the what I would call the systems biologists, who tend to look at networks and so forth, start talking with the structural biologists, who tend to look at the physicality of interactions. And I think that's starting to happen. It's certainly something that I'm very interested in and trying to get some work going in it. Yeah.
Any other questions? Yes, Carl.
I just have to make a comment, and that is that listening to what you just said, I thought of something that could be done with your technology that would have a profound effect on humans, on the way we look at the world. And that is that there's a book that was written originally by Mark Tashney on the genetic switch. And I was thinking, there is a place where they've actually tried to learn all the molecular events that occur when a virus decides whether it's going to kill the bacteria or integrate into the genome and stay there peaceably. And if that were illustrated this way, I think that would be dynamite.
Yeah, and I believe people are starting to do that, and we're interested in a variety of those same examples where you actually know kind of what makes a bacteria decide to do one thing or another. I'm sorry, I couldn't hear that. Oh, prions? That's an interesting question. I haven't done work on prions myself, although I have collaborated with people that are working on other types of plaque forming. So prions are one type of plaque forming or misfolding protein, but there are lots of others. Some genetic diseases there's one that involves transthyretin, and which exists both. It exists in the blood as a tetramer. 4 molecules come together, and it functions in the blood. But it can come apart and refold as a long fibrous structure. And people, it's a familial, there's a familial form of dementia that is based on this transthyretin. It also affects the heart. And so one of the things that we were interested in collaborating with a colleague at Scripps, Jeff Kelly, is to design a drug that would stabilize the tetramer so that it couldn't fall apart. And that's actually, I think, already gone into clinical trials. So these things do, these kinds of things are important. There's a colleague at UCLA, David Eisenberg, who's done structures of these. So prions are these peptides. And so they can form these kinds of structures. And I showed him these models. He wanted to have a model so he could kind of use them to kind of understand how these different fragments can come together. So yeah, we're very interested in that. I haven't, there's only so much time to work on a variety of things. So what I'd like to do is to try to get the technology out to people so that they can use it.
I forgot to give my name, Rudy Krutar. I'm a member of the society. The physicist who was attacking a zoological problem came up with the assumption that all elephants are perfect spheres. And he assumed that there was a room that was a perfect sphere with two elephants inside that half the diameter of the room, and spherical zebras around the contact between the two elephants. This is the Sanguka problem, and it happens that the centers of the two different kinds of spheres form a 3, 4, 5 triangle. You notice any other interesting numerological features with your packing?
The short answer is no. Yeah, but I can't say that we've looked. But I'm not sure what kind of numerological or geometrical things one might find. When you try to populate these cellular environments, they're very irregular. They're highly. I mean, you saw what these organelles like the mitochondria and so forth look like in that movie. And cells are just filled with all of these substructures that partition the space in a variety of different ways. And so I don't think that you're going to find, simple mathematical structures per se, although you might find interesting patterns. I think the patterns are abstractions of what's going on, and whether they have meaning or not, just like whether the spherical elephant has a meaning or not, depends on the questions that you're asking. Mike?
I think we're a little bit out of time, so it'll be time during the social hour to ask more questions. I'm assuming we'll be around for a while.
Yeah.
But let's thank Professor Olson. Yeah, one more thing for you this evening.
Oh.
You're not done yet.
I'm not done yet.
This is the announcement framed and signed by the members of the General Committee.
Oh.
Thank you for your...
Well, thank you
really
very
see
much.
you.
Thank you.
Thank you very
It's
much.
been a pleasure.
Thank you.